Hello students, have your seats. Today we are going to discuss regarding lysosomes. You know, lysosomes are one of the main component of endomembrane system. From last 3 sessions, continuously we are discussing on the endomembrane system. We have completed discussion on endoplasmic reticulums and Golgi bodies. This is the third component of the endomembrane system, which we have to discuss now. Before starting detailed discussion on lysosome, here I would like to share one of the important information with you. Listen students carefully. This institute always have been trying to impart information to you on each and every subjects with a innovative and new advanced form of techniques of teaching, so that you students grab information more and more and you may get success in your fields. In this regard, as a further step of advancement in the field of teaching style of this institute, recently our institute has started preparing videos of each and every classes imparted by different faculties, and we are uploading those videos on YouTube channel. The name of channel is the same, Topping Success Tutorials, which is the name of this institute. Here I would like to suggest you students, at your home every night you go through those videos so that you can revise the lesson which we are already taught same day in the classroom tutorials. Till the date, on cell biology, we have published 14 videos on YouTube regarding different topics of cytology, particularly on the cell biology. Still now, our man is preparing the video for this session. So you go through all those videos so that you can get benefited and you can save your time from the tedious book reading and going through the lengthy voluminous notes. Let us start discussion on the lysosomes. As you know, the lysosomes are one of the components of the endomembrane system. It is very tiny, minute, spherical, membrane-bound cell organelles, which are found scattered in the cytoplasm. They are very tiny in structure. Their size may vary from 0.23 to 0.5 mu or micron. You know about the micron? Micron, 1 micron is 1 millionth meter. You can imagine how they are tiny, how they are minute. Actually, they are electron microscopic. Only with the help of electron microscope we can see them inside the cytoplasm of eukaryotic cell. Now we will discuss regarding discoveries of lysosomes. There was a great scientist, namely Christian de Duhe, who first time discovered lysosomes. And not only he discovered, he also coined the name of this membrane-bound tiny cell organelles of eukaryotic cells as a lysosomes. But you should know here that this great scientist discovered the lysosomes in the animal cells. After him, there was another scientist, namely P. Martin. He discovered or recognized first time the lysosomes in the case of plant cells. Now we will discuss where the lysosomes are found and where they are not found. Normally they are found in all the eukaryotic cells as well as in plant cells and animal cells. But in animal cells they are frequently found. Their numbers are more in respect of plant cells. Now we will discuss where they are not found. They are not found in all of the prokaryotic cells as well as in human RBC, red blood corpuscles. Now we will discuss actually what this membrane bound tiny electron microscopic cell organelles is. This word, see here attentively, this word lysosomes is made up of two parts of word. First is lyso, lyso, and some. Lyso means digestive and some means body. The word lysosome is telling its own story. The body which pertains to digestion. That means certainly this cell organelles pertains to digestion inside the cytoplasm of the eukaryotic cells. Really, this is the fact. This cell organelles is called as digestive bag. Why? What is the reason? Here we should know. Because they contain more than 40 types of enzymes and are capable to digest most type of the materials which are found inside the cytoplasm. So they are called as digestive bag. Now we will discuss regarding origins of lysosomes. In last 3 sessions, we have discussed that all the 3 members of endomembrane system work together in well coordination and well association. In this context, you should know the lysosomes are originated by the Golgi bodies in association with endoplasmic reticulum. See here, I have drawn a separate diagram for showing you the origin of the lysosomes. See here, this is the simple endoplasmic reticulum and this is the cluster of Golgi bodies. You know, inside the cytoplasm, a smooth endoplasmic reticulum carry the synthesized materials like protein and lipids to the forming phase or cis phase, you may tell, that is the convex phase of the Golgi bodies. And there they bud off, particularly the vesicles of the smooth endoplasmic reticulum bud off those material which they were carrying at this phase. Thereafter, the system of the endoplasmic reticulum receive those material, and you know, the modification, the processing, and then packaging takes place inside the Golgi bodies, particularly in cisternae. And after processing, modification, and packaging of the materials, ultimately the modified form of the material are sent to the maturing phase or trans phase of the Golgi bodies. There, by the process of budding, the cisternae bud off and leave those modified form of the materials, and in the form of vesicles, and by this way lysosomes are formed, and then they are scattered in the cytoplasm in different destination and points in the cytoplasm. This overall process is in short form called as GERL. GERL stands for Golgi associated endoplasmic reticulum lysosome synthesis. You remember this term. This is very important on examination point of view, particularly for NEET point of view. GERL. Again I am repeating, GERL. This is the process for the synthesis of lysosome in the cytoplasm of all the eukaryotic cells. Again I am repeating this process and the origin of the lysosome. Lysosomes are synthesized in the cytoplasm of all the eukaryotic cells by the Golgi bodies in well association and coordination of endoplasmic reticulum, particularly the smooth endoplasmic reticulum. Now we will discuss again in context of why the lysosomes are called digestive bag. Just before I told you, because they contain number of types of enzymes, digestive enzymes. Specifically, you should know they contain more than 40 types of hydrolytic enzymes inside them. And with the help of those enzymes, they are capable of digesting all type of carbohydrate, protein, lipids, and nucleic acids which are found in the cytoplasm. Here are the some forms, you see, of the hydrolytic enzymes which are found inside the lysosomes. First form is the acid phosphatase. Second is acid ribonuclease. Third form is acid deoxyribonuclease. And fourth form is cathepsin. Fifth is sulfatase. Apart from these forms, there are also number of types, number of forms of the hydrolytic enzymes which are found inside the lysosome. Now we will discuss regarding certain examples of these hydrolytic enzymes. See here, I have written 3 examples of these forms of hydrolytic enzymes which are found inside the lysosomes. Some are lipase, proteases, carbohydrases. These are the examples of hydrolytic enzymes. Due to presence of number of hydrolytic enzymes inside them, they are capable to digest all the contents of the cells. This is the reason they are called as digestive bag. In next class, we will discuss on certain terms and points regarding lysosomes, like why they are called suicidal bag, why they are called demolition squad, why they are called housekeeper of the cell, how they are called garbage disposal system. On these terms we will discuss in next session. Apart from these terms, we will also discuss the various type of the function which are performed by the lysosomes. Again here I am repeating, as our man is preparing video of this session, and in evening today we will upload that video on YouTube channel. So at night you watch that video so that you can revise the lesson which just I taught you. Thank you very much.